Hello everyone, welcome back to my channel. I hope you all are doing well and you are preparing pretty well for your upcoming examination. So this video will particularly focus on the PYQs of GATE Biotechnology. So stay tuned and do watch the complete video to get insights of what can be asked and how to approach a particular question. If you like my efforts, you can hit the like button and do subscribe my channel for more such content. So the first question is, the relationship that involves the exchange of nutrients between two different species. In the question, a kind of relationship is asked in which it was said that exchange of nutrient is taking place in two different species for their mutual growth. It means it is on a beneficial note that both the species are getting benefit from it. They are mutually growing with that exchange of nutrients. Is it antagonism? No. Antagonism will not mutually benefit each other. So antagonism is cancelled out. Commensalism? No. Commensalism is also not so, because in this commensalism one is benefited and the other is neither benefited nor harmed. So is it parasitism? No. It is not parasitism, because parasitism is a relationship between two organisms in which one benefits at the expense of the other. So it is not parasitism. So eventually we have trophism. The correct answer. So basically syntrophism is also known as syntrophy or cross feeding, which is a biological phenomenon in which two or more species are mutually benefited for their growth when they cooperate to form a metabolic function. It means when they share their nutrient requirement with each other, they mutually grow, and this process is known as syntrophism. Also spell out syntrophism. Syn means together and trophism means nourishment. Now the second question. The second question is about which one of the following bacteriophage has a genome composed of single-stranded circular DNA. Single-stranded circular DNA. So it is the phage 174 which has the single-stranded circular DNA. So the correct answer for this question is option A. Now, depending upon the previous question, I have provided this chart because it can be asked in this year. This is basically the viruses, and they are classified on the basis of their genetic material, that whether they have DNA or RNA, and that too with envelope or non-envelope. So basically these are the viruses that infects the vertebrates. So DNA viruses with envelope are pox viruses and herpes viruses. And you have to remember that pox viruses have double-stranded DNA and the herpes virus have double-stranded DNA again. So accordingly, you have to remember that which of the following group of viruses has either DNA or RNA, and that too with an envelope or non-envelope. So the DNA viruses with no envelope in their structure is adenoviruses, papovaviruses and parvoviruses. So basically adenoviruses have double-stranded DNA and papovavirus has double-stranded DNA again, but parvoviruses has single-stranded DNA. You have to remember it because it will be so much confusing when it is asked together in the form of match the following format question. Because you all know that in GATE, match the following questions are frequently asked in 2 marks. So you have to remember it. Now RNA viruses with envelope. So RNA viruses are Toga virus, orthomyxovirus, paramyxovirus, rhabdovirus, coronavirus, which you all must be familiar with, flavivirus, filovirus, Munya virus and retroviruses. So all these viruses are RNA viruses, and that too with an envelope. And the RNA viruses with no envelope are picorna viruses and reoviruses. You have to remember only 2. So the trick to remember is you have to remember the exceptional thing, because अगर कोई चीज़ बहुत ज़्यादा होती है तो उसको हम याद रखने में मिस कर सकते हैं, उसे हम भूल सकते हैं. सो बेसिकली यू नीड टू डू दी एक्सेप्शन वाली चीज़ आपको याद रखनी है, या फिर जो थोड़ी बहुत चीज़ें हैं वो याद रखनी है. फॉर एग्जाम्पल, अगर यहाँ पे RNA वायरसेस विथ नो एनवलप में याद रखना है तो यू ओनली हैव टू रिमेंबर 2 थिंग, दैट इज़ दी पिकोरना वायरसेस एंड रियो वायरसेस, दैट दे हैव RNA वायरसेस विदाउट एनी एनवलप. बट अगर आप जाते हैं RNA वायरसेस विद एनवलप में याद रखने को तो आपको बहुत सारी चीज़ें याद रखनी होंगी. सो बेटर कि ऑड वन आउट वाली चीज़ें याद रखें, जनरलाइज चीज़ें वैसे ही याद रहती हैं. नेक्स्ट, एंड एन ईजी क्वेश्चन, an element that is present in a nucleotide but not in a nucleoside is, it is obviously the phosphorus which is present only in the nucleotide but not in the nucleoside. So the correct answer for this question is phosphorus. Now next question. Krebs, that is the tricarboxylic acid cycle, that is the TCA cycle, is dash pathway. Only an anabolic pathway, only a catabolic, an amphibolic, or a pyogenic pathway. So it is the amphibolic pathway. It means it has both anabolic and catabolic phases in its metabolism. So how it is anabolic? So when TCA cycle generates precursor for the biosynthetic processes, such as it form the fatty acid, amino acid. So on that note, it is anabolic, and it is catabolic in terms of when it oxidizes the acetyl group to produce carbon dioxide and reducing power. Now next question. All pseudogenes, you have to focus on the word pseudogenes, do not code for a dash. So basically pseudogenes are non-functional DNA segment that resemble the functional genes but are unable to code for proteins. बेसिकली क्या बोल सकते हैं pseudogenes को? Pseudogenes जो होते हैं वो नॉन फंक्शनल DNA सेगमेंट होते हैं जो कि पूरी तरह से एक जीन को ही रिजेंबल करते हैं बट दे आर अनेबल टू कोड फॉर ओरिजिनल प्रोटीन. सो इसमें से कौन सा ऑप्शन फिट बैठ रहा है? एज पर द डेफिनेशन आई हैव जस्ट मेंशन्ड, protein with original function. This is the correct answer because protein with altered function, RNA with coding sequence, RNA with regulatory function, this is not the correct one because pseudogenes क्या होते हैं? वो DNA segment ही होते हैं जो कि non-functional होते हैं and they are unable to code for any kind of protein. The correct answer for this question is all pseudogenes do not code for a protein with original function. Now one match the following in which so many diseases are asked along with its biological vector. First one is Chagas disease. So basically this disease is also known as the American trypanosomiasis, in which the disease is caused by the protozoan parasite known as the Trypanosoma cruzi. You must be aware about the Trypanosoma cruzi, which is the responsible or the causing agent of this disease. Now the vector that transmits that parasite or transfer that parasite into the host is known as the kissing bugs. Now the bug is scientifically known as the Redwood bug. It means P will be matched with the 4. Now the trypanosomiasis. So trypanosomiasis is caused due to the tsetse fly. So Kyoka 1. Leishmaniasis, the vector of leishmaniasis is sandfly, and for the yellow fever it is the mosquito who transmit the major causative agent. So as per the matching P4, Q1, R3 and S2, the right answer will be option 1. So through this easy matching you can gain 2 marks in GATE exam. So you should attempt the match the following question because they are easy and you can easily gain 2 marks. Now one more matching question in which different enzymes are mentioned along with its application. So the function of lipase is oil degradation. Basically it is an enzyme responsible for the degradation or hydrolysis of the oil. So pica 2. Now fikin. Fikin is responsible for the tenderization of meat. It means Cuca 4. Amylase is responsible for the generation of fructose or maltose syrup. It means it acts on the starch. Basically it hydrolyzes the starch into maltose. And glucosidase. Glucosidase is responsible for the hydrolysis of carbohydrate or basically the polysaccharides into oligosaccharides and monosaccharides. So glucosidase is for the oligosaccharide production and for the monosaccharide production. So answer will be option 2. Now one more question which is based on the Sanger DNA sequencing. Which one of the following is the basic principle of Sanger's DNA sequencing method? Chain termination by incorporation of dideoxynucleotide. Yes, this is the correct option. Chain elongation by incorporation of dideoxynucleotide. No, chain elongation तो हो ही नहीं पाएगा because incorporate क्या हो रहा है? Dideoxynucleotide हो रहा है. Now release of inorganic pyrophosphate. No. Chain cleavage by modification of dideoxynucleotide. No. So the right answer for this question is chain termination by incorporation of dideoxynucleotides. Sanger sequencing relies on the principle that the growing DNA strand is terminated by addition of the dideoxynucleotide. It means when you add the DDNTP, that is the dideoxynucleotide triphosphate, so the growing DNA strand will immediately terminate. And what is the reason behind it? So the DDNTPs basically lacks the 3-OH group, which is needed for the further elongation of the DNA strand, and due to the absence of this 3-OH, the growing chain terminates. Now one more important question which is confusing also. So you have to mention the correct function. Now the primase. So the function of primase is to synthesize the short chain RNA onto the DNA strand to initiate the replication process. So primase works on what? It works on the DNA dependent RNA synthesis. So P का क्या होगा? P का होगा 4. Now reverse transcriptase. So the reverse transcriptase act on the RNA dependent DNA synthesis. So Q का 3. Now RNA replicase. RNA replicase will act on the RNA dependent RNA synthesis, and DNA polymerase will definitely work on the DNA dependent. So the right answer for this question is option D. I hope you gain an insight of what can be asked and how to approach a particular question in GATE. So if you want more such videos, you can comment below. And thank you so much for watching.